Our method aims to generate heart field-specific cardiac progenitor cells from mouse embryonic stem cells.
This heart field reporter stem cell line allows for the high-throughput study of the mechanisms underlying congenital heart disease, providing a system that is amenable to genetic and pharmacological manipulation.
There are several chamber-specific congenital heart diseases. By recapitulating cardiogenesis in vitro, this protocol allows the study of disease mechanism and the development of future regenerative therapies.
This protocol can provide insight into how cardiac progenitor cells of different chambers are specified during heart development.
Begin by growing transgenic mouse embryonic stem cells in 0.1% gelatin-coated T25 flasks in 2I medium. When the cells reach 70 to 80% confluence, rinse the cultures with PBS and add 1 milliliter of trypsin per flask to dissociate the culture into single cells at 37 degrees Celsius for 3 minutes. When the cells have detached, neutralize the reaction with 4 milliliters of 10 FBS in DMEM and count the cells by an appropriate method. Dilute the cells to an approximately 3 times 10 to the 5 cells per fresh medium concentration and collect the cells by centrifugation. Then resuspend the pellet in 5 milliliters of fresh 2I medium for replating onto new 0.1% gelatin-coated T25 flasks. For CPC generation from cardiac spheroids, collect 2.5 times 10 to the 6 of the detached transgenic mouse embryonic stem cell sample by centrifugation.